Science behind the history of the microscope. In the 17th century, when the first microscopes were invented, there were several epidemic diseases that were poorly understood, such as the bubonic plague and smallpox. Scientists and physicians recognized the need for a tool that could help them better understand the causes and mechanisms of these diseases, and the microscope provided them with a way to observe and study microorganisms that were too small to be seen with the naked eye. Over the centuries, the microscope continued to evolve, with advancements in optics, materials science, and engineering leading to the development of more complex and sophisticated microscopes, such as the electron microscope and the scanning tunneling microscope, paving new paths of research and innovation. The first microscope. In the 14th century, Italy was the first country to produce spectacles, while in 1590, a father and son team of Dutch spectacle makers, Hans and Zacharias Janssen, invented the first compound microscope by using multiple lenses in a tube. This early microscope consisted of two lenses that were placed at either end of a tube and allowed for a magnification of up to 9 times. They discovered that by putting a lens at the top and bottom of a tube and looking through it, objects on the other end could be magnified. This invention was an important foundation for the future developments in microscopy. However, the device could only magnify between 3x and 9x. These first compound microscopes were limited in their ability to increase resolution, resulting in blurry images. Therefore, no significant scientific advancements were made for about a century. However, by the late 1600s, improvements to the lenses improved the quality of the image and magnifying power to up to 270x, leading to major discoveries. History behind light microscope. One interesting story behind the history of the light microscope is the discovery of cells by Robert Hooke in 1665. Hooke was a natural philosopher and an English natural scientist who worked for the Royal Society in London, and he was one of the first people to examine objects using a microscope. One day, he decided to examine a piece of cork, and he was amazed to see that it was made up of tiny, empty compartments that he called cells. Hooke's discovery was a major milestone in the development of the microscope and the study of biology, as it revealed that living organisms are made up of discrete, organized structures. In fact, the term cell is still used today to describe the basic unit of life. In 1667, Micrographia was published by Robert Hooke, featuring a plethora of detailed illustrations of various specimens that he had examined. Among these was a herbaceous plant branch that showed separate segments, which he also documented. These sections were called cells, and Hooke became known as the father of cellular biology. Hooke's discovery also paved the way for the work of other scientists, such as Antoine van Leeuwenhoek, who was the first person to observe living cells and bacteria under a microscope. Antoine van Leeuwenhoek, the father of the microscope. Antonie van Leeuwenhoek, a Dutch cloth merchant turned scientist, improved the microscope in 1676 to look at the cloth he sold. However, he accidentally made a groundbreaking discovery that bacteria existed, which opened up the field of microbiology and laid the foundation for modern medicine. The theory that diseases were caused by noxious air and unpleasant smells, known as the miasma theory, was contradicted by Louis Pasteur, a French scientist, who almost 200 years later established that bacteria were responsible for a multitude of illnesses. According to Kevin Elisieri, a microscopist at the University of Wisconsin-Madison, the discovery of bacteria was a significant event. He described it as huge and stated that it caused a lot of confusion about the cause of the illness. The realization that bacteria and other microorganisms exist in water was one of the greatest discoveries in history. The year 1677 marked an important discovery by Leeuwenhoek, who was the first to identify human sperm. A medical student had provided him with the ejaculate of gonorrhea-infected patient for microscopic examination. Upon observation, Leeuwenhoek detected minuscule creatures with tails that he termed animalcules. He later discovered these same organisms in his own semen. Although he published these groundbreaking findings, their true significance was not understood by scientists for another 200 years, as was the case with the discovery of bacteria. In the late 1800s, Walter Fleming, a German scientist, made a significant discovery in cell division, which eventually helped in understanding the growth of cancer. This finding would not have been possible without the use of microscopes. Despite the limitations of the original microscopes used by Hooke and Leeuwenhoek, their fundamental structure of two lenses connected by tubes remained significant for many years. Before proceeding further, please subscribe to the channel and click on the notification icon so you would get notified when we upload the next video. The discovery of the electron microscope. The history of the electron microscope is also a fascinating one. The first electron microscope was invented in 1931 by German physicist Ernst Röske and electrical engineer Max Knoll. Röske was interested in finding a way to observe specimens in greater detail than was possible with light microscopes, and he realized that using electrons instead of light would allow for much higher magnification. Röske's first electron microscope used a magnetic lens to focus a beam of electrons onto a specimen, which was then magnified and projected onto a fluorescent screen. The first images produced by the electron microscope were low resolution, but they still provided a level of detail that was far beyond what could be seen with a light microscope. Over the next few decades, researchers continued to improve the design and capabilities of the electron microscope. In 1938, Röske and his brother Helmut developed a transmission electron microscope, or TEM, which uses a thin specimen that is illuminated by a beam of electrons that pass through it. The electrons that pass through the specimen are then focused by magnetic lenses onto a detector, which creates an image. In 1965, researchers at Cambridge University, including Ernst Röske's student Sir John B. Pendry, developed the scanning electron microscope, or SEM. This type of microscope uses a beam of electrons that is scanned over the surface of a specimen, and the electrons that are scattered back are detected to create an image. Today, electron microscopes are used in a wide range of scientific fields, including material science, biology, and nanotechnology. They're capable of producing images with resolutions that are thousands of times higher than those produced by light microscopes, and they have helped researchers uncover new details about the structure and function of cells, molecules, and materials. Röske's principles still form the basis of modern electron microscopes that can magnify up to 2 million times. The mass market for microscopes developed in the 20th century, with China becoming a major supplier, leading to more competitive prices and wider availability. New light sources and boom stands have improved versatility, and the digital microscope is the latest invention. Scanning probe microscope. The research achievements of G. Binning and H. Rohrer at the IBM Zurich Research Labs in Switzerland sparked a global interest in scanning probe instruments. SPM is a type of microscope that uses a physical probe to scan the surface of a sample at the nanometer scale. The probe interacts with the sample's surface, and the resulting data can be used to create an image or map of the surface. One interesting fact about SPM is that it can be used not only to create images of surfaces, but also to manipulate and control them at the atomic and molecular levels. This has opened up many possibilities for nanotechnology and materials science research. Another fascinating application of SPM is in the field of biophysics, where it has been used to study the structure and behavior of biological molecules such as DNA and proteins. SPM has also been used to create high-resolution images of individual atoms and molecules, providing valuable insights into their behavior and properties. Similarly, the STM, a special kind of SPM, works by using a tiny, sharp tip that is brought extremely close to the surface of the sample being studied. A voltage is then applied between the tip and the sample, which allows electrons to tunnel between the two. By measuring the tunneling current, researchers can create a three-dimensional image of the surface of the material being studied. Tip-enhanced Raman spectroscopy, or TERS. Tip-enhanced Raman spectroscopy, or TERS, is a cutting-edge analytical technique that combines the power of Raman spectroscopy with the spatial resolution of scanning probe microscopy, or SPM. It uses a sharp metallic probe, typically a gold or silver nanoparticle, attached to the end of an atomic force microscope, or AFM tip, to enhance the Raman signal of molecules in close proximity to the tip. TERS is capable of achieving spatial resolutions on the nanometer scale, enabling researchers to study the vibrational spectra of individual molecules and materials at the submolecular level. This makes it an incredibly useful tool in the fields of chemistry, material science, and biophysics, where understanding the behavior of individual molecules and their interactions is critical. Perhaps the most exciting thing about the history of the microscope is that it's a testament to human curiosity, ingenuity, and our unending desire to explore and understand the world around us. So next time you look through a microscope, take a moment to appreciate the incredible journey that led to its creation and the countless discoveries that have been made possible because of it. Please leave a comment if you have any suggestions for the next video. Also, if you enjoyed watching the video, please hit the like button and subscribe to the channel for more videos like these. Thank you for watching.